Psilotum Introduction. It is most widely distributed genus of class Calypsida having more than 100 species. The most common and worldwide distributed species is Psilotum nutrum. It is commonly known as whisk fern. Habitat. The plant body is perennial and slender shrubby. It is commonly grow in humus-rich soil in tropical and subtropical regions. Some species grow as epiphyte. Node may be terrestrial in habitat and growing on the exposed rock crevice. Vegetative morphology. The plant body is rootless and leafless sporophyte and consists of dichotomously branched underground rhizome and upright aerial axis. Rhizome. It is covered with scales and bare rhizoids. Any branch tip of rhizome turn up to produce an aerial axis by apical growth. Aerial axis. The aerial shoots are green, upright and slender. The aerial axis may be cylindrical at the base, furrowed in the upper parts but somewhat flattened with 3 longitudinal ridges at the top. The basal part is smooth but the distal part bears small scaly appendages and syningia. The aerial axis performs photosynthesis. The appearance of aerial axis is xerophytic. However, plants grow in moist conditions. Internal structure or anatomy. Internal structure of the aerial branches. Internally the stem is differentiated into following structures. Epidermis. It is the outermost layer and consists of a single layer of cells. Cell wall of these cells is highly cunetinized. Stomata are present in the epidermis but do not have subsidiary cells. Cortex. Cortex is extensive and divisible into 3 regions. Outer region. This region lies directly beneath the epidermis and consists of chlorophyllous cells with intercellular spaces and contains chloroplasts and starch grains. These cells are called chlorenchyma. Middle region. This region composed of sclerenchymatous cells and without intercellular spaces. These tissues provide mechanical support to the plant. Inner region. This region composed of parenchymatous cells and without intercellular spaces but contains starch grains. Endodermis. Endodermis is present inner to cortex. Endodermis is formed of a single layer. These cells have the characteristic Casparian strips or bands on the radial walls. Pericycle. Pericycle is present inner to the endodermis. It consists of a single layer of parenchymatous cells. Stellae. Parenchymatous cells form the ground tissue of the stellae. Pith is present in the center. At the extreme base, the stem is protostellic but in the middle region, the stellae type is siphonostele. Internal structure of the rhizome. Internal structure of rhizome is closely related to that of aerial branches except following points. The epidermal cells give rise to rhizoids. The outer region of cortex contains intracellular endophytic fungus, mycorrhiza. The cells of middle cortex contain starch grains. The stellae is protostellic. Life cycle of Psilotum. Sporangia. Syningium. Asexual reproduction. Psilotum is homosporous and spores are produced within the sporangium. At maturity, many dichotomously branched aerial shoots become fertile and produce trilocular sporangia called syningia at the tip of very short axis. Development of the sporangium. A group of superficial cells initiate the development of each sporangium. It divides into primary wall initial and primary sporogenous cell. The primary wall initial further divides to produce the 4-5-layered sporangial wall. The innermost layer is tapetum. Primary sporogenous cell takes part in the formation of sporogenous tissue or archaeosporium. 1/3 archaeosporial cells enlarge and become spore mother cells. The remaining cells disintegrate to form mucilaginous liquid. This liquid provides the nourishment to the developing spores. Each spore mother cell divides meiotically to form 4 spores. Wall of spore becomes 4-layered. The outermost layer epispore splits to form 4 bands. These bands separate from the spore wall on drying. These bands are called alaters. These alaters help in the splitting of the sporangial wall and dispersal of spores. Germination of spore shocked phase. The spore germinates on suitable substratum and produce gametophyte. Sex organs are produced on the upper side of gametophyte. Sex organs. Sexual reproduction. Sex organs are scattered over the surface of gametophyte. Generally, Antheridia are more in number than Archaegonia. Antheridium shocked face. Each antheridium is produced from a superficial cell that divides to produce outer jacket initial and inner androgonial initial. Or the jacket initial divides to produce wall of the antheridium. The wall has a triangular opercular cell at the top. Or the androgonial initial divides to produce a mass of androgonial cells. Or each androgonial cell divides to produce two androcytes or antherozoid mother cells. Or each androcyte is changed into an antherozoid. The mature antherozoids come out by the lifting of the opercular cell of wall. Archaegonium shocked face. Archaegonium also develops from a superficial cell that divides to produce an upper neck initial and a lower central cell, or the neck initial divides to produce a neck. Neck is composed of 4 vertical rows of cells, or the central cell divides transversely to produce an upper neck canal initial and lower ventral cell, or the neck canal initial produces a row of 2 or 3 neck canal cells. The ventral cell divides to produce a large egg or oosphere at the base and a smaller ventral canal cell. Fertilization, or the ventral canal cell and the neck canal cell of mature Archaegonia disintegrate to form passage for the anthrozoids, or several anthrozoids enter the Archaegonia. But only one of them fuses with the egg, or the fertilized oosphere develops a wall and becomes the oospore. Development of the sporophyte or the oospore divides mitotically to form embryo that later divides into an upper epibasal cell and a lower hypobasal cell. Or the hypobasal cell divides to produce a foot. Or the epibasal cell divides to produce aerial branches and rhizome of sporophyte.